So the plan is, when we start looking at this assignment, is the page that I'm showing you right now on the screen is ultimately going to go on the back page or the back of this number one. Okay, and I'll explain those instructions here in a little bit. We'll kind of go through, but that's the ultimate goal. Is that when you take a look at, again, what I have up on the screen, I'm kind of talking the seventh period here as well. But you guys, you're going to end up cutting these out. Okay, and the solid lines are what you're going to end up cutting. That's how you're going to be able to know what you're cutting out. Okay, and ultimately what's going to happen is they're going to get put right into the individual boxes, right, that you're going to see on this page right here. All right, and like I'm showing up on the screen itself. So as we read through, okay, it says the cells below belong to an organism with a diploid number of 2n equals 6. So if I have a diploid number of 2n equals 6, what does that mean in a haploid number? It means 3, right? Because, again, you got 2n versus n, right? Okay, you're going to have 3 pairs. In this case, we have, right, just 3. There's no pairs whatsoever, okay? So the cells are in various stages of mitosis and meiosis, right? And count the number of chromosomes in each cell and label it 2n or n, right? Then you're going to cut out each of the cells so that's on its own rectangular piece of paper, match up any cells that are in the same stage of division, and then place them all on the proper place in the lab sheet, okay? So the ultimate goal is you're going to go through, first step I would probably tell you guys to do is determine whether in diploid or haploid state, okay? Next, all right, you can either write this down or cut them out, right? But I wouldn't probably cut them out until you absolutely know where it goes where, because then you're going to have losing pieces and so on down the line. But you're going to have to go through and determine, right, is this in prophase, metaphase, anaphase, telophase, or interphase, okay? And then you also need to determine, is this cell in mitosis, meiosis 1, or meiosis 2, okay? Then, again, as we said, as you cut them out, you're going to put them basically on this sheet. Mitosis. Here's interphase, prophase, metaphase, anaphase, telophase, and so on down the line. And there's 1,000 ways to skin a cat when we start going through this, meaning that there's multiple ways that you can do this, right? In that you could, one, cut them all out, find all the prophases, find all the metaphases, find all the anaphases, find all the telophases, and so on, and then split them up from there. You could also go through and try to find, well, these are all my mitosis, these are all my meiosis 1, these are all my meiosis 2s, and then split them up from there. Okay, I don't really care how you do it. You can just write them all down, and then when you cut them out, place them in the proper place, all right? And when you do place them in the proper place, you either staple, you glue, or tape. I don't really care what kind of method you use, all right? It's just you're going to end up having them to stick so that way you can hand them in to me, and I can make sure that what you did is correct, okay? Now, a couple things about this. As we go through, 2n versus n, okay? Again, if we're looking for diploid versus haploid, what number of chromosomes did it tell you was in the diploid state? How many chromosomes do we find in a diploid cell? What does it tell you up here? Right on the screen. It tells you 6, right? 2n equals 6. 2n is diploid. Okay? Which means haploid, again, has what number?
3.
That's 3. So, for example, if we look at this cell right here, okay? I got to kind of remember what I'm trying to do here. Okay? If I'm using this cell, looking at this cell right here, okay? Right? What does that cell, or what stage is that cell in? Right? Is it a diploid or is it a haploid stage? Okay, and why would you say diploid? Yeah, you got 6 chromosomes there, right? There's 6 X's. So I would go through and I would label that one, well, 2N. Okay, I'd put that down. Okay, what about this guy right over here? What would you say, is it diploid or haploid? It'd say be haploid, right? Okay, because there's only 3 X's. Okay, how about this one right here? Diploid, right? Why do we know? Why do we say diploid? Because there's 6 X's. Okay, what about this one right there? What would you say, diploid or haploid? Realizing it hasn't divided yet, right? So if it hasn't divided yet, you still want to do what? Count the total number of chromosomes, right? And so again, what do we count? 1, 2, 3, 4, 5, 6. It'd be diploid in number, okay? So I think that one should be pretty easy. Now, it goes through a skin cell. Well, is a skin cell diploid or haploid? It's a diploid, right? Because the only cells that are haploid are going to be the sperm and the egg, correct? Okay. I take a look, a reproductive cell. It even tells you, hey, this one is diploid, right? Here's some haploid ones, okay? Now, the next step, okay, and I don't care what your next step is, but you're going to want to determine, let's just go through this. You're going to want to determine interphase versus prophase versus metaphase, anaphase, and telophase, and they also include cytokinesis in on this, okay? So, again, we go back to the three structures. We go back to the nucleus, we go back to the chromosomes, and we go back to the spindle fibers as a way to acknowledge what phase they are in. So if we go back to this original cell that we started out with up here in the top left-hand corner, you guys said it's diploid in state. Okay, now would you say it's an interphase, prophase, metaphase, anaphase, or telophase? Okay, why would you say prophase? Chromosomes are there. What are we still seeing surrounding the chromosomes? Yeah, the nucleus is breaking up, but it's still there. Exactly right. Okay, exactly right. Chromosomes are visible. Nucleus is still there. So I would say this would be in, again, a prophase, right? Okay, how about this guy right over here? Upper right-hand corner. Okay, what phase would you say that would be in? I hear two different answers. I hear prophase and interphase. Say it again. Interphase? I like that one. And why would we probably choose interphase over prophase? I know the nucleus is starting to break up. I get that part. But what can't we see? There's no chromosomes and no spindle fibers, right? There's none of that going on there, right? So I would probably throw that as an interphase. Okay. How about like this cell right there? What would you probably say that bad boy is in? Metaphase, right? Because where are they all at? They're lined right down the center. Okay, so you guys can figure that part out. Now, the tricky part here, though, is we need to determine, okay, again, are they in mitosis? Are they in meiosis? Are they in a meiosis 1 or meiosis 2? Okay, so here's what we're going to do. Okay, I'm going to give you guys a little bit of help today. Can I draw on that? I can draw on that. Okay, we're going to give you guys a little bit of help, all right? And I have over on the board, right, basically a kind of a flow chart that you're going to use to determine whether it's in mitosis, meiosis, or meiosis 1 or meiosis 2, okay? And that one that I have is sitting right here. It's this guy right there, okay? Now, here's where we're at. So we're going to start out, first question, we could actually do this one of two ways. We're going to do it the way I have up on the board. First question we're going to ask is it a diploid number? Okay, so we're going to go through and you should ask every cell, is it diploid? Now, what's nice about this is that if that cell is not diploid, okay, it can only go one place. It can only be part of meiosis 2 because those cells are all haploid, okay? So I would put down no right here. It's not diploid. It would be meiosis 2. Okay. Now, obviously, on the other side, I would have, well, yeah, they are diploid. And if they are diploid, now we can break these actually up into 2. Okay. And the 2 we can break them up to is going to be whether it's mitosis or it's meiosis 1, right? So I'm going to put down mitosis and I'm going to put down meiosis 1. So the cells we find in mitosis and in meiosis 1 are all diploid. Okay? The cells we find in meiosis 2 are haploid. But now we got to be able to determine, okay, so what's the difference? That doesn't help me, basically to separate the mitosis and meiosis 1. So now the question comes up is, well, what is the defining factor? What is the actual factor that separates those two, right? And when you think about it, it's all based on how they line up in the center of the cell during metaphase or even pairing up in prophase. And we already said that in meiosis 1, what do we end up getting? They will do what? They will pair up in homologous pairs, right? In mitosis, I'm going to put down just no HP. Okay? So if I go back to this original cell, we've already said that this guy right here is diploid, right? We've already said that it is in prophase, but I still need to determine, okay, what process it's in. Well, diploid, yes, right? Now I jump down over here. Now I'm going to start looking for, are there homologous pairs, right? And when I look, do we see them paired up at all? No, they're not. So which would tell me that this right here is in mitosis, right? Yeah. Here would be an example right over here of that exact same cell, but it's in meiosis 1 because it's diploid in number and they are what? They're paired up, right? Exactly.
So they have to be lined up to be lined.
Well, they have to be paired up. Because in the case of like prophase, you may still see there just may be X's, but they are together, right?
Like 2 and 1.
Be something like that right there. Yeah, like, I mean, something like this. You're going to end up seeing a total of 6 cells, right? Or 6 chromosomes, excuse me, but they're all big, they're paired up, right? Where if it's just mitosis, they're not paired up, right? Okay. The other part that you start looking at is like during metaphase, right? You either have got 6 that are going straight down in a line. That would be mitosis because they're not paired up, or you have 3 pairs of chromosomes, 3 pairs of 2. That would tell you that it is in meiosis 1. Okay? Yeah. Okay. So pretty similar, pretty simple there. Now, the other part that I'm going to make up to you guys are basically reach out. Why can't I get rid of my pen? Where's my little guy at? Okay. Why did it just go away? I don't know why. I can't even back out of there. Can I go there? Where you at? All right, who are my technological advanced individuals in here? No idea? Okay. Let's go there. I'll go back to it. Classroom. Whoops, go Google Drive. Oh, and I still got my pen there. What is going on? Well, okay, all right. I'm going to have to explain this. Otherwise, I can't get that figured out. All right. So the one part I want to explain is going to be on that back page. Okay. And again, people in 7th period, make sure you guys are listening, is where you're going to place those little squares, those rectangles at. Notice that in mitosis and meiosis 1, right, they're the same sized squares, right? What are you noticing about meiosis 2? It's what? It's bigger, okay? Well, if you remember right, in meiosis 1, what happens? We're actually taking one cell and we are forming 2 cells. And in meiosis 2, we're taking those 2 cells and we're forming 4 cells out of that. So what you're going to end up having over on meiosis 2 is you'll end up having 2 cells that you're going to place in that box, eventually leading to a division of 4 cells, okay, on top there. All right. So now, like I told you guys in this class specifically, I would probably start on the back page today just because I'm here. I can maybe help you out a little bit more. I think the front page is pretty self-explanatory, and you should be able to go through that no problem on Monday. I would also tell you, do not start cutting these out until you're absolutely ready to paste them or staple them or whatever you're going to do, just so you don't lose them from basically from now until Monday. So if you just want to get stuff written down today, cut them out, put them out. I don't really care. That's up to you guys. Okay, just make sure that you're not losing this. I don't have a lot of extra copies of this. Okay, other than that, you guys can work together, right? Try to get this done, try to get this completed. You'll get Monday to work on it as well, so it's not like it's a huge rush by any means.